Thank you so much for having me today to talk to you about our research. So my group is based at the Manchester Fungal Infection Group here at the University of Manchester. And our research focuses on understanding how the cells that are lining our lungs, the respiratory epithelium, are able to shape either directly or indirectly the immune response to inhaled fungal spores. In particular, we are interested on how these cells ingest and eliminate fungal spores, how and when this process is important to help us clearing everyday exposure to fungal spores, and why and how this is not happening efficiently in patients which are predisposed to fungal infections. So, for example, COPD sufferers. I probably don't need to stress to this audience that fungal diseases are indeed a big problem for human health. Fungal infection cause more than 2 million human deaths a year, and this is as many as TB or malaria. But they are associated with much higher mortality than TB and malaria, which are diseases that perhaps we hear more often of. And the importance of fungal diseases has been broadly recognized more and more from worldwide initiatives such as that one today, but also by the recent release of the fungal priority pathogen list by the World Health Organization. So this is a list that it's basically ranking pathogens that cause acute systemic fungal infection. They pose a serious risk of morbidity or mortality in humans, and they are currently presenting drug resistance or other management challenges. And what it's important to notice is that with the exception of candidiasis, a lot of these fungal infections initiate in the airway following inhalation of fungal spores. And here you can see some examples of these beautiful yet little spores. Among these, Aspergillus species are the fungi that are most often cultured from the airways of chronically ill human beings. And Aspergillus fumigatus cause the majority of human diseases. Although Aspergillus spores are a common component of what we breathe, they are not the most prominent of the spores entering our human airway. So they've got specific features that make them very good for being to cause disease. So nature did not design Aspergillus fumigatus to be a pathogen of humans, but it's likely that things like the small size and thermotolerant nature of these spores promotes their survival in our lungs, and these results in different disease outcomes, which are depending on the host status. And although the highest mortalities are associated with the most rapidly progressive invasive syndromes, the population at risk of chronic and allergic diseases are larger. And among the risk factors for developing aspergillosis is COPD. So COPD, chronic obstructive pulmonary disease, is a condition that is characterized by persistent and irreversible limitation of airflow through the lungs. And although this list here is not meant to be presenting with all the studies, I'm here showing you some of the most recent studies that are showing how there is an association between COPD and aspergillosis, with a significant number of individuals with COPD being at risk of developing aspergillosis. Diagnosing aspergillosis in COPD patients can be challenging, and the actual prevalence might be underreported. This could be due to factors such as geographical location, diagnosis criteria, and patient populations. But what we are really interested in understanding why there is this association between COPD and aspergillosis. Now, what is known so far is that COPD patients have higher rates of Aspergillus fumigatus colonization than healthy people, and there are probably several reasons for this. This could be because the lung damage and tissue remodeling, constant tissue remodeling observed in COPD sufferers due to chronic airway inflammation creates a structurally and immunologically suitable environment for Aspergillus fumigatus spores to lodge within the lungs. And this is further worsened by the impairment in mucociliary clearance. So this mechanism that brings the spore up for them to get eliminated once we inhale them the excessive mucus production, which makes the airways very sticky, but also dysfunctional phagocytic clearance. So in COPD, innate immune cells, such as these cells here, macrophages, are unable to uptake and kill intracellular Aspergillus fumigatus as efficiently as their healthy counterparts. So you can see here, healthy macrophages would take up the spores and clear them, but this is not happening as efficiently as COPD. So I quite often think about macrophages as this Pac-Man in the video games from the 80s, that they go around patrolling our lungs and eating what they find and eliminating what it's not good for us. But how about airway epithelial cells? So airway epithelial cells are always there covering our lungs. And actually, if we think about the number of cells present in the airways, alveolar macrophages only constitute 3 to 5% of the cells in the alveoli. So they are unlikely to be the first cell type that are encountered by inhaled spores. And then if we also think about all the models where macrophages and other immune cells like neutrophils have been depleted, yes, these models show that these cells are very important in clearing the fungus that we inhale, but they are not able to do that without the help of something else. And we study airway epithelial cells because we believe that airway epithelial cells are something that it's coming to help innate immune cells. So the contact with airway epithelial cells and Aspergillus fumigatus is extensive, and it's likely to be prolonged. And actually, as you can see here, a unifying feature of all the Aspergillus-related diseases is the damage to the pulmonary epithelium. So the host pathogen interaction at the alveolar interface is a crucial event for the establishment of disease. The spores arrive, germinate, and penetrate and damage the tissue. But what is known for other respiratory diseases caused by bacteria, viruses, it's also that this interaction here, it's crucial for the containment of disease. And our research argue that in the majority of the cases, epithelial activities are actually helping to protect the Aspergillus exposed host. We have developed a lot of methodologies to look at how airway epithelial cells are able to internalize Aspergillus fumigatus spores and kill them. In our case, we have developed these strains that are fluorescently labeled, so they are red fluorescent, so that we can visualize when they are taken up by epithelial cells. And here you can see these are epithelial cells that, based on this staining, have got Aspergillus fumigatus in red or yellow inside. But actually, when the fungus is on the outside and only on the outside, they get stained with also this blue dye here. This allows us to measure how our models in the lab are able to take up fungal spores. And you can see here that this is a time-dependent process. It's a time-dependent process that pretty soon leads to the killing of what has been internalized. So here you can see something on the outside, it's growing, it's not killed, but something on the inside, it stays as a spore. So it seems like it's killed. And we wanted to understand more about this process, and we wanted to understand if this is aberrant in disease. So our next question was, does COPD significantly impact the uptake and the killing of fungal spores? And we were able to have access to human cells, to test this hypothesis. And what we found was that indeed, COPD cells are not only able to internalize the Aspergillus fumigatus spores, but they seem to internalize much more. They're stuffing themselves in fungus. But what is not happening after this is that the spores are killed. So the COPD cells you can see here have a much lower ability to kill what they have internalized. suggesting that aberrancy of the ability of epithelial cells to respond to spores might contribute to susceptibility to aspergillosis in COPD sufferers. So we know that uptake and killing of fungal spores by epithelial cells, it's a potent way of helping in antifungal defense following exposure to Aspergillus fumigatus. And we know that these processes might become compromised in patients with COPD, leading to heightened susceptibility to fungal infection. So understanding how epithelial cells, it's very important for us because it could have a clinical importance, and it could inform the development of much needed novel antifungal therapeutics. So my group is now focusing very hard to try to understand how this process works and how it doesn't happen as efficiently in COPD. So the first thing that we wanted to understand is we know, I've shown you in the histology at the beginning, that the fungus, when it's in the lung, goes from being a spore to then germinate and form this ivy that penetrate the lung tissue. We wanted to understand if any of these forms is taken up more abundantly by airway epithelial cells. And to do that, we produce these different, we call them morphotypes, basically shapes of fungus. These are the different developmental stages, which we can then challenge the epithelial cells with doing infections of epithelial cells in the lab. And what we found was that indeed there are specific morphotypes, specific shapes of stages of the fungus, which are taken up more abundantly by epithelial cells. But why? What is on the surface of these morphotypes that is making them so tasty for the airway epithelial cells? So we used a panel of stains that bind to different components of the cell wall and of the fungus. And we found that one of them in particular, called concanavalin A, it's binding to those stages that are most abundantly taken up by epithelial cells. Now, concanavalin A binds to something on the surface of the fungus, which is called mannose. And we thought, well, can we, if mannose is important and concanavalin A is blocking mannose, is interacting with mannose, can we use either concanavalin A or mannose to block uptake of the fungus by epithelial cells? And indeed, what we found was that both of them, when we used them in our infection of epithelial cells, there was a decrease. We're able to inhibit the uptake of Aspergillus fumigatus by airway epithelial cells. So that showed to us that something that is binding to the mannose on the fungal cell is helping the fungus to be taken up by airway epithelial cells. And we thought about a lot of, we tested a lot of receptors that we know are binding mannose. A few didn't give us any result, but then we found that one of them in particular was quite a good candidate in our experiments. And this is surfactant protein D. So surfactant proteins are secreted in the alveoli, specifically by alveolar epithelial cells. So the cells that we're actually using in our experiments in the lab. And they bind to mannan, but they're also known to bind to Aspergillus fumigatus already. And here you can see that they are specifically secreted by the airway epithelial cells. What we found was that when we were blocking, either using a specific antibody against SPD or genetically engineering these cells to eliminate SPD activity, the cells were not able to take up the spores as efficiently as when they were having a functional SPD copy here in white. So we have discovered that SPD is a novel driver of Aspergillus fumigatus uptake by ECs. And there is a lot that we are currently doing. We are in the process of evaluating the role of SPD during infection. And also we are looking at it as a potential molecule involved specifically in the defects that we've seen in COPD. At the moment, we haven't been able to test it with COPD cells, not yet. This is currently ongoing. But what we found very reassuring is to actually see that there are a lot of studies already available that for different aspects are showing an association between surfactant protein, the misregulation or genetic variation with COPD and its severity. So we are looking at this, we have as a very promising target, and we are pushing really hard to focus on this. So how can this help us in the clinic? So as I mentioned before, we think that it would be very important to understand the mechanism of uptake and killing by airway epithelial cells, because understanding how this works would help us to understand if defects in these processes are associated with increased susceptibility to aspergillosis. But it could also provide us with a good predictor of the risk of developing aspergillosis and a diagnostic marker for the risk of developing aspergillosis. And so finally, I just wanted to say thanks to the people who are involved. This is the entire Manchester Fungal Infection Group here at the University of Manchester, and you would have seen some of the people that conducted the work. Sebastian here and Patrick in particular here. Were in my group, and yeah, they've conducted the majority of the work that I've presented today. Thank you.
Has anybody got some questions? There's one question in the Q&A that we can start with. Do you think that spores can live inside the macrophages after they've been engulfed? And will they be relapsed after you use the treatment to epithelial cells? So
I think actually in the presence of specific pharmacological treatment and immunosuppression, that is actually what a good possibility, so that they would survive while they are in the macrophage or in the epithelial cell. The way that I see it, though, is that the epithelial cells come first in the interaction. So if anything, looking back at the way that the question was put, I think it's more likely that if there was an incomplete killing, the epithelial cell will serve as a reservoir for whatever it's coming afterwards. I don't know if that answers the question.
I think so, yeah. I've seen some amazing fluorescence microscopy videos at a previous conference. I think it was Candida, where it had been engulfed, and the Candida, it was trying to expand with the sort of the microtubules, and the immune cell, it was just like folding it up like 10 poles. They were kind of having this real fight in real time. It was amazing.
Yeah, I mean, intracellular lifestyle of pathogens, and in particular fungal pathogens, I mean, I find it very fascinating, because the differences as well for different fungal pathogens, for different cell types. So we know that the thing that probably you mentioned in Candida, you've got specific behaviors within a macrophage and within neutrophils, and they are both cells that are designed to kill the fungus, right? But depending on the nutritional environment that the fungus encounters once it's inside the specific cells, because these are different, then the infection is different. It's all very fascinating in my view, but just because I like it a lot.
It does sound very cool, yeah. Hugh, you've got your hand up. Would you like? to ask something.
Hi, Margherita.
Hello.
I agree with you. They are cool and fascinating talk. You talked about COPD and macrophage activity or airway epithelial cells. Did you discriminate between current smokers and former smokers in your COPD group, and did that make a difference? I often bang on about smoking is the most important thing to stop if you have ABPA.
Yeah, absolutely. So unfortunately, our cohort of patients that we could test so far is quite limited. So we don't get the luxury of, in a way, if you want to say it, we don't get enough cells that we can investigate, enough donors that we can investigate that. Quite often they are all smokers, but one of the things that we want to do is actually, because as well, another thing that I think would be important to do is actually look at the COPD severity and see whether there is, some stratification depending on the severity of COPD. So that's something that we want to do. It's also true that when we actually get information from, because some of the donors we can buy commercially, and the information that we get is quite limited. But quite often we only have smokers.
I'd be very happy to get you some samples of if you need them.
Lovely, that'd be great. That'd be great. Thank you.
Yes, smoking is one of the things that we talk about in the support groups, and it's one of those questions where I think people who have smoked, they feel so much shame over either having smoked or they can't quit yet. If anybody is thinking about coming to one of our monthly meetings, we're going to talk next month about how help available to stop smoking if you want to, because it's National No Smoking Day on the 16th of March. Actually, I had a oh, sorry.
Beth, I just had a comment really. So if I just to interpret this simply, because people might not quite have followed the pace of that, what you've essentially done is detect how our bodies eliminate pathogen, eliminate fungal canidia. And because you now understand that, and you understand that a receptor can promote internalization, which then goes on to destruction of the fungus. Putting it very simply, if we could give patients who don't have enough receptor genetically or because of environmental things like smoking, if we could replace that receptor, we could restore internalization, and that could effectively be a medication to help reduce the amount of fungus in somebody's lungs.
Yeah, that's absolutely well put. And actually, one thing to add on that, thinking about the specific candidate that we have now identified, it's already known that SPD is protective against invasive and allergic aspergillosis in models, in infection models. And SPD is, as far as I know, I mean, I'm not a clinician, but it's already used as a therapy. So, for example, it's given to preterm babies. So it's already used as a therapy. So yeah.
Nice. Okay, that's really, really encouraging. It's another route to trying to eliminate or reduce all kinds of aspergillosis and presumably other fungi as well. Cool. Thank you very much. Very nice talk.
Thank you.
Can I ask a quick question?
Yeah.
Do we have any idea what it is about this, like what components of the cigarette smoke is causing it? Like, could you get the same mechanism from a different irritant, like traffic pollution or things people get exposed to in workplaces, or is it something about cigarettes particularly?
I actually don't know. That's a very good question. What we are trying to do actually is looking at the effect of smoke extract directly on our models. So we've linked with the hospital to be able to work on these. But in that case, if you think about, for example, looking at specific components, that might be something that we can investigate. But I don't know the answer at the moment.
I'm just thinking, because like smoking, the rates are the rates of smoking have kind of been coming down over the last few decades, but air pollution is a lot more on the radar.
Yeah, absolutely.
I wonder if we'll see.
Absolutely, that's a very good point.
Oh, brilliant! That was. We've just got Chris joining. That was absolutely fantastic talk. Thank you so much. I might have to watch it again on YouTube later to get all the details. But yeah, that's.